Gene therapy, in short, is the therapeutic use of both genes and shorter, non-coding DNA and RNA sequences in order to restore the correct form of a mutant gene, obtain its expression, or modify the activity of individual genes. First, there are ex vivo therapy and in vivo therapy. In vivo therapy, genetic material is introduced directly into the patient's cells in their body. Ex vivo therapy involves taking cells from a patient, modifying those cells outside the patient's body, and introducing genetically altered cells back into the patient's body. It is obvious that ex vivo therapy is safer because you have full control of the cells taken from the patient, and in case of any problems, the cells can be reharvested and the cells can be genetically modified again in a different altered way. The second division of gene therapy separates it in terms of the effect it produces, and so we can distinguish gene supplementation therapy, suppressor or suppression gene therapy, and anti-cancer gene therapy. Supplementary gene therapy consists of delivering a therapeutic gene to cells, which is a correct copy of the damaged or inactive form of the gene. In this case, the material delivered to the cells is usually cDNA, created on the basis of an mRNA template in the process of reverse transcription. In order to deliver a transgene to cells, a suitable vector must be used that allows the transport of the introduced genetic material across the cell membrane. Vectors used nowadays have a limited capacity, therefore the aim is to compensate from the introduced genetic material as much as possible. mRNA-based cDNA consists of exons only and it's devoid of introns. It means non-coding elements that do not carry any important genetic information. Removal of the introns significantly reduces the size of the introduced transgene and facilitates its transmembrane transport. Suppression therapy consists in inhibiting the overexpression of a mutant gene at the level of transcription or translation. For this purpose, DNA and RNA antisense, ribozymes, siRNA, shRNA, microRNA, and aptamers are used. Antisense numbers are from 20 to 30 nucleotides in length, the sequence of which is complementary to the mRNA, the DNA sequence of the gene, or its promoter. Inhibition of the synthesis of unwanted proteins can proceed in two ways, either by associating an antisense to a selected pre-RNA sequence, disrupting the assembly, or by associating an antisense with a mature mRNA, blocking access to the ribosome, or creating a heteroduplex degraded by RNases. Ribozymes are molecules capable of specific enzymatic cleavage of mRNA molecules. Specific mRNA cleavage may impair or prevent translation of the corresponding proteins. Aptamers, on the other hand, are RNA molecules with high affinity for compounds. For example, they can block polymerase by incompetent inhibition by blocking the access of the template to the active site. siRNA, microRNA, and shRNA, in short, silencing gene expression by degrading mRNA. The differences between the individual molecules are present in the table. Cancer is the result of the accumulation of many changes in the genetic material, therefore the repair of all damaged genes would be ineffective, and therefore most often attempts are made to selectively eliminate neoplastic cells by acting directly using suicidal or pro-apoptotic genes, or indirectly stimulating the immune system or using anti-angiogenic therapy. Anti-angiogenic therapy works by blocking the expression of the blood vessel growth factor VEGF. As we know, cancer cells proliferate very quickly, so they need a very large amount of nutrients that they obtain from the blood. By blocking VEGF, the growth of blood vessels within the tumor is inhibited and the cancer cells cannot get a lot of nutrients. As a consequence, the growth of neoplastic tissue is inhibited. What usually all types of gene therapy have in common is the use of vectors whose task is to effectively introduce genetic material into cells and protect the transgene against degradation. There are two types of vectors, viral and non-viral. Introducing genetic material by means of viral vectors is called transduction, while introducing genetic material by means of non-viral vectors is called transfection. Currently, the gene therapy transgene is delivered to cells most often by viral vectors due to their relatively high transfection efficiency. The word should be emphasized here relatively, because it is still about 20 to 30%. Viral vectors use the natural ability of viruses to infect. Some genes responsible for virulence and replication are replaced with a transgene. Viral vectors are widely used in gene therapy, although their use is associated with a number of inconveniences and risks, such as a small space for an insert or often the transduction of only dividing cells. The real problem, however, is their safety. Many such vectors are immunogenic and have a severe immune system response. Almost all viral vectors, characterized by long-term expression of the introduced transgene, integrate the therapeutic gene into random sites, enabling the formation of inert mutations. Due to so many inconveniences and dangers, an alternative in the form of non-viral gene transfer is being sought. Non-viral transfer relies mainly on the use of plasmids as carriers of therapeutic genes. The advantage of plasmids, the circular molecules of genetic material over linear molecules, is that they can be easily multiplied together with the transgene in bacteria, and that the plasmids are resistant to the action of endonuclease, that is, they are characterized by greater stability. In the present slide, we can see a diagram of a typical expression plasmid vector. The expression cassette comprises a transcription factor binding promoter and RNA polymerase, a transgene, and a polyadenylation signal. ORI sequences ensure replication in bacteria, F1 ORI, and eukaryotic SV40 ORI cells. Antibiotic resistance genes allow the selection of bacterial cells containing the vector, and the CoE1 gene regulates the replication cycle of coli bacteria, as this type of bacteria is most often used to multiply vectors. The plasmids themselves are introduced into cells by various methods, as the plasmids itself does not have the ability to penetrate cell membranes due to its size, negative charge, and hydrophilicity. The transmembrane transport of plasmids is possible due to condensation with molecules of lipid, polymer, nanoparticles, or dendrimers, or the use of physical factors such as sonication or electroporation. However, the main disadvantage of non-viral gene transfer is low efficiency, which is influenced by factors such as degradation of carriers, problems with the release of complexes from endosomes, problems with stability in the cytoplasm, and poor penetration into the cell nucleus.